Somewhere in the open ocean, a pod of sperm whales is sleeping, not lying on their sides, not resting on the seafloor, standing, vertical in the water column, like enormous grey monoliths, their blunt heads near the surface and their bodies hanging down into the dark below. A research vessel off northern Chile once drifted into the middle of exactly such a pod, engine cut, crew silent. And the whales didn't move, didn't flinch, completely inert, suspended as if someone had switched them off. The crew had to physically make contact before the animals stirred. That incident helped catalyze a 2008 study that tagged 59 sperm whales across multiple ocean basins and confirmed what that accidental encounter suggested. Sperm whales sleep vertically, in synchronized bouts of roughly 10 to 15 minutes, spending about 7% of their day this way near the surface. Groups of 5 to 7 animals drifting together in eerie parallel, breathing without waking. It's one of the most alien images in nature. And it's the calm before what this animal actually is. Because if one of those sleeping giants were to turn its head and direct its sonar at you, a diver suspended in the water nearby, the resulting click would arrive at your body at somewhere between 230 and 236 decibels, measured in water. That figure is referenced to a different pressure baseline than airborne sound, so it doesn't translate directly to the decibel scale you use to describe a jet engine. But at the scale of tissue physics, the distinction doesn't offer much comfort. Acoustic bioacoustician Bertel Merle and his colleagues at Aarhus University confirmed source levels up to 223 decibels using a 5-hydrophone array off Andenes, Norway in 2000. Subsequent research by Feiss and colleagues put the on-axis peak-to-peak maximum above 235 decibels. What reaches your body at close range wouldn't just blow out your eardrums. The pressure wave at that intensity would travel through your water-rich soft tissues like a concussive punch delivered to every cell simultaneously. Not sound as you normally experience it, but a physical force, a full-body impact. No documented case exists of a sperm whale click killing a diver, and under typical encounter conditions, the risk is considered negligible. But the physics of what that sound is at the source isn't in dispute. To understand how this animal generates it, you have to go inside the head. And what you find there rewrites the whole story whalers told about it. The sperm whale is an extreme marvel of biological engineering, possessing bizarre anatomical adaptations that allow it to dominate the deep ocean, weaponize sound, and sustain a highly complex culture. Every major system in this animal, its diving physiology, its acoustic anatomy, its neurology, its social architecture, is a solution to a problem no other creature on Earth has solved at this scale. Begin with the environment itself. Below roughly 1,000 meters, sunlight ceases to exist entirely. The water temperature drops toward freezing, and the pressure, at 2,000 meters, it's already 200 times greater than at sea level. Hydrostatic pressure increases by approximately 1 atmosphere for every 10 meters of seawater depth. At 3,000 meters, you are looking at roughly 300 atmospheres of pressure, around 4,400 pounds per square inch, bearing down on every surface. That is the world the sperm whale descends into on a regular foraging dive. The deepest confirmed instrumented dive for a sperm whale sits at approximately 2,250 meters, with some tracking data from Watkins and colleagues reaching just over 2,000 meters in the 1990s. Educational sources cite potential maximums approaching 3,000 meters, with the appropriate hedge that these are estimates of capability, not tagged records. The deepest instrumented mammalian dive on record belongs to Cuvier's beaked whale, not the sperm whale. But even at its confirmed operating depths, this animal is descending into a place where the pressure alone would crush any sealed chamber of gas, any rigid structure, any system not specifically engineered to survive it. Typical foraging dives last between 45 and 90 minutes. A male killed in 1969 after surfacing from a dive had been under for 117 minutes. The outside estimate for a maximum dive approaches 2 hours. An air-breathing mammal shouldn't survive this. Full stop. The first adaptation is in the ribcage. As pressure increases with depth, the lungs compress and the alveoli, the tiny gas exchange sacs, collapse. This isn't the whale choosing to collapse its lungs in any conscious sense. It's a passive, mechanical response to escalating hydrostatic pressure, built into the flexibility of the ribcage anatomy. But the consequence is precisely targeted. When alveolar gas exchange shuts down at depth, nitrogen stops dissolving into the bloodstream. Decompression sickness, the condition that kills human divers who ascend too quickly after breathing compressed gas at depth, becomes effectively impossible. The whale doesn't surface slowly to avoid the bends. It surfaces whenever it wants, because the gas that would cause the problem was never absorbed in the first place. The lungs compressed, the nitrogen stayed where it was, and the blood remained clean. The oxygen that drives the dive doesn't come from the lungs at all. It was loaded into the animal at the surface and stored chemically throughout the body's tissues, which brings us to the muscle. Myoglobin is the protein that gives vertebrate muscle its red color. It binds oxygen and releases it during aerobic activity, functioning as a local oxygen reserve within the muscle fiber itself. Human femoral muscle carries roughly 0.6 grams of myoglobin per 100 grams of wet tissue. Marine mammal muscles have been documented carrying concentrations up to 10 times that, and in deep diving cetaceans, the ratio is particularly extreme. The sperm whale's locomotor muscles are so saturated with myoglobin that they appear a dense, almost chocolate dark red, shading toward black. One French-language biological summary describes cetacean flesh as almost black, and attributes it directly to myoglobin saturation. Formal dissection reports tend toward dark red-brown, but the visual shift is real, and the cause is the same. An oxygen store so concentrated it changes the color of the tissue. This isn't an incidental quirk. Myoglobin in deep-diving mammals evolved a structural modification that enabled these extreme concentrations. The protein became more electrically charged on its surface, causing individual molecules to repel each other rather than clumping together, which would otherwise impair their oxygen-carrying function at high densities. A 2013 paper in Science traced this adaptation across mammalian diving lineages, showing that net surface charge in myoglobin scales with maximum dive duration across species. The physics of protein interaction was modified over evolutionary time to allow the oxygen battery to run bigger. The result is a system where a sperm whale loads its muscles with oxygen at the surface like a pre-charged battery, then draws that reserve down over the course of a dive that can last nearly 2 hours. Combined with elevated blood hemoglobin concentrations and specialized vascular structures called retia mirabilia, networks of intertwining arteries and veins that help manage oxygen and pressure, total oxygen stores in a large adult sperm whale can exceed 40 to 50 liters. The heart rate slows during the dive in a reflexive bradycardia, and blood is preferentially routed to the brain and heart while peripheral circulation cuts back. The sperm whale doesn't hold its breath the way a human does. It becomes a different physiological system entirely. It descends into the dark, runs on stored oxygen, and hunts. What it is hunting, and how it finds prey in complete blackness at 2,000 meters, requires understanding the structure of the head, which is where the story gets genuinely strange. Roughly 1/3 of the sperm whale's total length is skull and forehead. In a large adult male, this can reach 5 meters. Whalers in the 18th and 19th centuries discovered that the inside of this vast head contained a waxy, yellowish oil that solidified and turned white on contact with air, resembling reproductive fluid. They called the structure the spermaceti organ and assumed they understood what it was for. They were wrong on the biology and wrong on the function. The spermaceti organ isn't an oil reservoir. It's an acoustic machine. Inside the forehead, there are two primary fat-filled structures. The upper one is the spermaceti organ itself, a large, fibrous, oil-filled cylinder covered by an outer case of extremely tough connective tissue, fibrous cables as thick as a man's thumb, running in an interlocking mesh. Below it sits the junk, a layered structure of spermaceti-filled compartments separated by connective tissue, built directly on the bony rostrum of the skull. At either end of the spermaceti organ are air sacs, the distal sac at the front of the snout and the frontal sac at the back, pressed against the curved amphitheater of the skull. Clicking begins with air. The whale closes its blowhole and forces pressurized air through the right nasal passage, a channel that runs below the spermaceti organ toward the tip of the snout. At the end of that passage, just before the distal sac, is a pair of thick, black, lip-like valve structures, the phonic lips, known in French anatomical literature as the museau de singe, the monkey's muzzle. When air drives across these lips, they vibrate rapidly and produce an acoustic pulse. In Norris and Harvey's original 1972 NASA technical report, the foundational paper on sperm whale acoustics, they proposed that most of this sound energy escaped forward directly. Later work by Merle, Madsen, Zimmer, and others overturned that assumption. The 2005 three-dimensional beam pattern study by Zimmer and colleagues, mapping the full multi-pulse structure of sperm whale clicks using recordings from known aspects, confirmed what became known as the bent horn model. Most of the click energy doesn't travel forward from the phonic lips. It travels backward, posteriorly through the spermaceti organ toward the skull. At the back of the head, it strikes the frontal air sac, which functions as an acoustic mirror, reflecting the pulse forward. The redirected energy then travels through the junk, whose layered, compartmentalized architecture functions as an acoustic lens, focusing the sound into a tight forward beam. The primary click exits through an anterior terminal acoustic window at the rostroventral edge of the junk, the pointed front bottom of the whale's head, as a narrow, highly directional pulse. Merle's 2001 post-mortem experiment provided direct evidence. His team injected frequency-modulated pulses into the distal sac and junk of a recently deceased sperm whale stranded at Rømø in the Danish Wadden Sea, less than an hour after the animal was last seen alive. The recordings showed a repetitive, decaying reflection pattern, reminiscent of the multi-pulse click peculiar to sperm whales, matching the theoretical pathway precisely. Sound bounced back and forth between the air sacs, generating the characteristic multi-pulse train that researchers recorded in the open ocean. The anatomy performed as predicted. The directionality index of the resulting beam is roughly 27 to 35 decibels, meaning the on-axis intensity is tens of thousands of times greater than the sound radiating off-axis to the side or rear. The beam angle is approximately 10 degrees or less. At full output, this is a precision instrument, focused to a narrow cone, firing at source levels that reach 235 to 236 decibels peak to peak on the acoustic axis. That instrument then points into the abyss and listens for what comes back. Somewhere far below, in complete darkness, in water near freezing, something is listening too. Giant squid, Architeuthis ducks, are among the largest invertebrates on the planet. Recent confirmed size estimates place female individuals up to approximately 13 meters in total length, including tentacles, with mantle lengths around 5 meters. Their arms and tentacles are lined with suckers, each sucker surrounded by a chitinous ring whose edges are serrated with fine, sharp teeth. Sucker ring teeth, technically, a protein-based biopolymer embedded in the ring structure. In Argentine specimens, the largest sucker rings on the arm carry up to 52 of these sub-equal teeth around their circumference. When a giant squid grabs prey or resists being grabbed, these toothed sucker rings dig in and hold. They aren't hooks. Rotating hooks are a feature of the colossal squid, Mesonicoteuthis hamiltoni, a different species entirely. The distinction matters because the circular scars found on sperm whale skin, documented from strandings and whaling records across multiple oceans, are produced by the toothed sucker rings of large squid, not by hooks. those scars are the most direct evidence of what happens in the deep. No camera has ever recorded a sperm whale actively hunting a giant squid at depth. The encounters occur below 1,000 meters in total darkness, at pressures where no current remotely operated vehicle stays for long. Everything we know about the predator-prey relationship between sperm whales and giant squid is assembled from indirect evidence. Squid beaks recovered from whale stomachs, circular scar patterns on whale skin consistent with large sucker rings, and one precise finding from 2013 that cuts straight to the evolutionary heart of the relationship. Dan Erik Nilsson and colleagues published a paper in Current Biology that year, analyzing the extreme eye size of giant and colossal squid. Giant squid eyes can reach 27 centimeters in diameter, the largest eyes of any living invertebrate. The study's conclusion was that eyes this large can't be explained by the need to resolve fine detail or detect bioluminescent prey. At the depth where these squid live, the optimal eye size for detecting small light sources would be far smaller. But very large eyes are optimal for detecting the large, faint, luminescent disturbance created by a large animal moving through bioluminescent water from a distance of perhaps 20 meters or more. The only predator large enough to trigger this kind of disturbance in the squid's habitat, the study argued, is the sperm whale. The giant squid evolved its extraordinary eyes specifically to see this predator coming. The predator shaped the prey's anatomy across evolutionary time, and the evidence is written in the squid's face. Sperm whale clicking during a hunt begins with wide spacing between pulses, regular echolocation clicks at intervals of half a second to 2 seconds, providing long-range detection of prey concentrations, potentially at distances exceeding 15 kilometers in some estimates. As the whale closes in, the interclick interval shortens into a rapid train called a creak, analogous to the terminal buzz of echolocating bats closing on a flying insect. The creak provides rapid updates on a fast-moving target at close range. For years, researchers proposed an additional weapon in this sequence. The acoustic stunning hypothesis suggested that a sperm whale could deliver an intense burst of sound at close range to debilitate prey before capture, a sonic knockout blow in the dark. The physics, at close range and on axis, made it seem plausible. But in 2016, Face and colleagues published a detailed analysis in scientific reports using DTAG acoustic tag data from sperm whales actively foraging. During the terminal buzz phase, the final attack, the whales didn't increase their click intensity. They reduced it by 1 to 2 orders of magnitude. Prey exposure levels during the final approach were more than an order of magnitude below estimated debilitation thresholds. The study's title was explicit. Sperm whale predator-prey interactions involve chasing and buzzing, but no acoustic stunning. The clicks are a precision echolocation instrument. The weapon is biological engineering, not sound. The battle scars on whale skin suggest the squid doesn't surrender easily. The toothed sucker rings leave their circular impressions across the flanks of adults, sometimes in dense clusters consistent with multiple tentacles applied simultaneously. The animals arriving at the surface from these dives carry the record of encounters no one has ever witnessed. Now consider what is driving all of this. The navigation, the long-range echolocation sweeping the dark water column, the three-dimensional acoustic tracking of fast-moving prey in conditions that would defeat every human instrument without power. Behind the wall of spermaceti and acoustic lensing, inside a skull that the spermaceti organ has effectively reduced to a supporting basin, there is a brain. It weighs between 7 and 9 kilograms. Multiple anatomical sources converge on a mass range of 7 to 9 kilograms for adult males. For comparison, the human brain weighs approximately 1.3 to 1.4 kilograms. The African elephant brain reaches about 6 kilograms. The sperm whale brain, by absolute mass, is the largest of any animal known to have lived. Inside a 50-ton animal stretching 18 meters, it looks absurdly small. A 9-kilogram brain in a body that large is proportionally modest. The encephalization quotient, which measures brain mass relative to body mass, isn't particularly impressive by mammalian standards. But in absolute terms, no known animal, living or extinct, terrestrial or marine, is confirmed to have carried more neural tissue. Fossil brain cavities are rarely preserved with sufficient fidelity to make confident comparisons across extinct lineages, but the available record consistently places the sperm whale at the top. What does that brain do? The animal it serves is matriarchal, organized around groups of approximately 10 to 12 related females and immature males, coordinating across the open ocean over timescales of years. The brains of these animals are running something that looks very much like culture. Codas are rhythmic patterns of clicks produced during social interaction, not the echolocation clicks used during deep dives, and not the slow, deep clicks males produce for long-range communication, but a third acoustic register entirely. Sequences of interclick intervals that function as social signals between closely associated females and young. In 2001, Luke Rendell and Hal Whitehead published Culture in Whales and Dolphins in Behavioral and Brain Sciences, a paper cited over 1,300 times that made the case for cultural transmission in cetaceans and treated sperm whale codas as central evidence. 2 years later, they published a study in Proceedings of the Royal Society B, specifically documenting vocal clans, groups of sperm whales whose coda repertoires were distinct from other groups and best explained not by genetics, but by social learning. The dialects were transmitted through association, not inherited through DNA. The 2022 paper by Hirsch and colleagues in PNAS identified 7 distinct sperm whale vocal clans across the Pacific Ocean, each characterized by its own identity codas. The paper described these codas as a form of symbolic cultural marking, acoustic badges that identify membership in a social group. Calves born into one clan learn its dialect from the females around them. If those calves are separated from their clan and encounter members of another, the dialects differ. 27 coda types have been catalogued from the repertoire of Dominica sperm whale pairs alone. Across the Eastern Caribbean, Pacific, and Atlantic, dozens of distinct clan dialects have been identified. Leitao and colleagues published preprint evidence in 2024 of social learning operating across symbolic cultural barriers. Whales apparently adopting or modifying elements of codas through interaction with members of different clans. This isn't human language. The structural rules of coda sequences, whether they possess anything analogous to syntax, whether the order of interclick intervals carries combinatorial meaning, remain scientifically unresolved. Equating sperm whale codas directly to human linguistic structure would outrun the evidence. But they are learned, they are transmitted across generations, they are specific to social groups, and they function as identity markers across an ocean. That is a form of cultural inheritance. Generations of information encoded in click patterns passed from mother to calf across decades of association in the open sea. And then, for roughly 3 centuries, human beings hunted this animal at industrial scale. Sperm whales were the most commercially valuable cetaceans in history. Their spermaceti oil lit the streets of 18th and 19th century cities, lubricated the machinery of the Industrial Revolution, and formed the basis for one of the most profitable maritime industries the world has ever seen. By the mid-19th century, when Herman Melville published Moby Dick, inspired by documented incidents in which sperm whales attacked and sank whaling vessels, the hunt had already stripped populations across every ocean basin. The 21st century industrial phase was worse. Soviet whaling fleets deliberately falsified their catch reports, concealing the true scale of the slaughter from the International Whaling Commission for decades. Hal Whitehead's 2002 population work produced the primary estimates of pre-whaling versus current sperm whale numbers, documenting a substantial reduction from historical levels. The IUCN currently lists Phyceta macrocephalus as vulnerable. Ongoing threats include ship strikes, plastic debris ingestion documented in stranding cases, noise pollution that disrupts echolocation and communication, and climate-driven shifts in prey distribution. What was lost isn't only numerical. Each sperm whale clan carries a dialect. Each dialect represents generations of social learning, of mothers teaching daughters which click patterns mean what, of cultural information accumulated over timescales that dwarf individual human lifespans. When whaling killed individual whales, it killed members of these clans. When it reduced populations to a fraction of their original size, it likely erased dialects entirely. Cultural lineages with no surviving speakers, click patterns that were practiced for generations and then simply stopped, because the whales that carried them were gone. The sperm whale is an animal that spent millions of years evolving lungs that compress at depth to prevent poisoning its own blood. Muscles so saturated with oxygen-storing protein they turn the color of dark chocolate. A head filled with one of the most sophisticated acoustic instruments in biological history, and a brain large enough to run a generationally inherited culture across the open ocean. It did all of this in the dark, at pressures that would kill everything else, hunting animals large enough to leave permanent marks on its skin. We nearly destroyed it for candles.